Hello everyone, welcome to my course on microbial biotechnology. We are starting a new module today which is on food production involving microorganisms and their products. So, in this lecture we will be discussing about fermented food, their foundations and the principles involved in the process. So, we have broadly two sections. The first section deals with the historical and cultural perspective of fermented food. Then we have the second section which deals with the scientific principle of fermentation. So, we begin with a definition of fermented food. So, here you can see fermented radish which is displayed in a store. So, we are using the definition provided by Campbell Platt in his book The Fermented Foods of the World, a dictionary and guide. So, where he has defined fermented food as those food that have been subjected to the action of microorganisms or enzymes, so that desirable biochemical changes cause significant modification in the food. So, if you look at the biological and the chemical perspective of fermented food, biologically fermented foods result from the action of beneficial microorganism as defined by Campbell. So, these include the bacteria, yeast or molds that metabolize nutrients in the food, enhancing preservation, flavor and nutritional value while promoting probiotic activity. Fermented foods are defined by the chemical transformation of organic compounds, primarily sugars, into acids, alcohols and gases through microbial metabolism. This process alters the food's flavor, texture and preservation properties. So, extending on these definition, we have certain raw materials on which certain organisms and enzymes act to give a product which is the fermented food. So, we may have enzymes like chymosin, and then we can get the microorganism from natural spontaneous structure cultures, or also we can have commercial pure cultures. Now, certain conditions like aerobic and anaerobic environment and certain chemicals, salt, nitrates, sulfides and a combination or even including sugars, they interact with one another and then they change the whole nature of these raw material, and thereby we get transformed products which have better flavor and aroma, texture, and also the shelf life is increased. And of course, we need to be careful about certain allergens and then also the phytates. Now, the food that is produced at the process of these process is not only giving us food, it is also giving us certain bioactive molecules like peptides, phenolics, etc. And also it may give other valuable products like lactate, acetate, ethanol and carbon dioxide. So, overall this is a transformative process of fermentation. Raw materials are fermented under specific conditions, creating desirable foods with novel health promoting compounds while eliminating the harmful ones as shown in this diagram. So, let us look into the history of fermented food, how old it is. It has been known for thousands of years and various civilizations have used traditional fermented technique. For example, if you see the Mesopotamia, Tigris and Euphrates rivers, the inhabitants of these very old civilization, 6000 BCE there is evidence of lactic acid fermentation for the cheese production. Probably this is the first known use of fermentation coinciding with animal domestication and agricultural development. Then we have the earliest known alcoholic fermentation reported from China and Georgia, which is a beverages made of rice, fruit and honey, which is around 5000 BCE old. And then we have the development of wine production from grape juice in Egypt around 4000 BCE. So, there is an advancement in alcoholic beverage production, as you can see in this case. Then we have evidence of fermentations of beverages from Babylon, and then pre-Columbian Mexico, and then Sudan ranging from around 3000 to 1500 BCE, And then of course, also the elaboration of beer using barley in Sumeria. This will actually move a little bit up over here around 1800 BCE, and this has been supposed to be the first beverage which is distinct from the alcoholic beverages mentioned earlier. Then we have in the modern time, roughly around 1857 AD, we have Louis Pasteur, who has for the first time chemically explained the fermentation and provided the scientific understanding of fermentation as a living process. Then we have others like Edward Buchner, who advanced the understanding of fermentation mechanisms. Then we have first industrial production of yogurt by Isaac Carasso in 1919, then discovery of nicin, an antimicrobial product of lactic acid bacteria, and then publication of list of microbial food cultures recognized as safe grass by 2012, which is important from the point of view of establishment of safety standards for fermentation cultures in food and beverages. So, over historical timeline, we can see around 10,000 BC there is spontaneous dairy fermentation, and then around 7000 BC ancient Chinese civilization begin to brew, and then the 2000 BCE the cucumber is pickled in the Tigris Valley. The Chinese use moldy soybeans as an antibiotic around 500 BCE. Then fermentation of tea leaves was first documented in Chinese civilizations around 200 BCE, and then between 500 to 1000 AD cereal legume fermentation becomes globally known. And then in the modern times, Louis Pasteur work, as we have already discussed, and then the identification of bacterial strains in 1900s by Oleg Machkinov, a Russian bacteriologist, who discovered the bacterial strain Bulgarian bacillus in fermented milk, later classified as Lactobacillus acidophilus. Now, this is a very important microorganism from the point of view of probiotics, and by 1970s the first ingestible probiotic was commercialized, and the field has been progressing quite rapidly over the years. So, some of the notable figures and their contributions to fermentation, both food and beverages, we can see here Louis Pasteur, which we already mentioned. He advanced the understanding of fermentation, including ethanolic, lactic, butyric and acetic types, by isolating lactic bacillus and proving it converts glucose to lactic acid. So, he introduced the concepts of aerobic and anaerobic life, revealing that some microorganisms can thrive without oxygen, challenging previous fermentation theories. So, he was the first to have a patent on purified yeast in 1873. The patent included new fermentation methods for beer and described the yeast as free from disease causing germs. Significant in debates about patenting natural products, as the US patent office cited this patent to justify isolating genes. Pasteur's patents helped control his biotechnological inventions, while his vaccines were compensated by the state rather than patented. So, Pasteur's brewing process, basically he studied beer and wine making, patented a process improving beer quality. Previously, wort was boiled and cooled by exposure to air. Pasteur's method involving cooling wort in closed vessels by spraying water on the outside, introducing special yeast post cooling to prevent contamination from the wild yeast. Then we have Emil Christian Hansen. He gave us the isolation techniques to isolate single yeast cells, enabling pure yeast strain propagation and preventing contamination in brewing. So, this is a huge impact on brewing and revolutionized the brewing industry, improving beer quality and consistency. Quickly adopted the major breweries like Carlsberg, which still is a very successful brand. He basically tried to reduce spoilage and made brewing more reliable by eliminating the unpredictability of the mixed cultures. His techniques still influence modern brewing with ongoing advancement in yeast and fermentation technology. Now, what is the importance of fermented food? So, the question is basically why fermented food when we have fresh food available. Number one is it helps in the preservation. There are times in the year in certain geographical localities which may be very extremely cold or maybe extremely hot, or it may be very heavy rainfall, food may not be available. So, it is important to preserve food for long term use. So, historically food fermentation extended shelf life by creating acidic environments and compounds that inhibit harmful bacteria, yeast and molds, preventing spoilage and foodborne illnesses without modern refrigeration. Enhanced flavor and aroma was also one of the reasons why people preferred certain food in fermented form. Fermentation breaks down the organic complex carbohydrates and proteins, releasing organic acids, alcohols and esters that create unique flavors and aromas, as seen in the tangy and umami rich profiles of foods like cheese, yogurt, sauerkraut and kimchi. Then fermentation food also provides nutritional benefits. For example, certain food like yogurt, kefir provides probiotics, while certain fermentation microorganisms break down nutrients for better absorption by the body. Then fermented food also have reduced anti-nutrients, compounds that hinder nutrient absorption by breaking them down and increasing the bioavailability of nutrients. And another thing is the reduction of toxins. Fermentation can partially break down cyanogenic glycosides, for example, in cassava, which releases toxic cyanide when consumed, and reduce or eliminate some of the food toxins. And it has huge significance from point of view of culture and culinary traditions. Fermented foods are integral to the culinary traditions of many cultures, essential to regional cuisines, connecting people to their heritage and enhancing the diversity of global culinary experiences. And one of the important thing is waste reduction. So, the food, if fresh food is not consumed, I mean the food will be wasted. So, fermentation can transform surplus or imperfect produce. Okay, certain in quality control, certain foods may be sorted out to be of not high good quality. Those can be actually transformed into fermented food as well as fermented beverages, and it produces the flavorful and nutritious food, thereby reducing waste. So, all these helps us in sustainability. So, fermentation is a sustainable food preservation method that requires minimal equipment and energy, unlike energy intensive processes like canning or freezing, making it eco friendly. Then there is the significance of artisanal and creative foods. Fermentation encourages experimentation and creativity in the kitchen, allowing unique flavors and textures to emerge through variations in fermentation times, ingredients and techniques. Let us now briefly look into the role microbes play in the fermentation process. The fermented food microbial ecosystem is actually very complex. This involves not only lactic acid bacteria and Saccharomyces cerevisiae, but also other microorganisms like coagulase negative cocci, corniform bacteria and non-Saccharomyces yeasts. These organisms interact and sometimes compete with the primary fermenting microbes, adding complexity to the fermentation process. Traditionally, microbiology relied on culture dependent methods which involve isolating and cultivating microorganisms. However, these methods have limitations as do not always reflect the true diversity of microbial communities. Many species, especially those in low numbers or with specific growth conditions, may not be represented accurately. Culturing conditions in the lab often fail to replicate the natural habitat of these microbes. So, we have culture independent methods which have emerged, offering a more comprehensive view by analyzing the microbial DNA and RNA directly from the food matrix. These methods do not require culturing, allowing for the detection of all microorganisms present, even those that cannot be grown in vitro. This shift has revolutionized the study of microbial ecosystems in food, particularly with the advent of next generation sequencing technologies like pyrosequencing, in which allows for the detailed analysis of microbial communities, including rare species. Recent studies using both culture dependent and independent approaches have expanded the understanding of fermented products from dairy, meat and vegetable sources, revealing new insights into microbial dynamics. Let us now discuss about the various types of fermentation that happens in the production of food as well as beverages. Briefly, they can be listed as shown in this figure as lactic acid fermentation, alcoholic, acetic acid, malolactic, butyric acid, and mixed fermentation. We will be discussing some of these in little detail here, and some of the others in the lecture where we will be dealing with fermented beverages. So, lactic acid fermentation is a metabolic process where sugars are converted into lactic acid by lactic acid bacteria, essential for producing various fermented food. It includes acid fermented vegetables like kimchi, sauerkraut, and then acid fermented cereals and beverages, traditionally the cereal beers, then acid fermented seafood and meat mixtures. So, the microorganisms which are involved in lactic acid fermentation are primarily the species of lab, including Lactococcus, Lactobacillus and Streptococcus. They provide health benefits, enhances the digestibility of food, improves gut health through probiotics, and can extend the shelf life of products by inhibiting spoilage organisms. And they have culinary usages in East Asia. These fermentation methods enhance the flavor of savory dishes, rice wines and plant-based beverages. So, here we can see various fermented foods on the left side of the table, and particular lactic acid bacteria involved in their production. We will not go into complete details of here, the information is available, you can consult this table at any point of time. So, basically for dairy products, butter and buttermilk, yogurt and cheeses, you have L-lactis, then you have subspecies lactis, then you have Delbrueckii subspecies bulgaricus, then you have acidophilus, then you have mesenteroides, and also Streptococcus thermophilus. And for fermented meat and fish products, we have the LB sake and then LB plantarum. Then we have here the P. pentosaceus and various undefined labs. So, we have in plant products like sauerkraut, olives, cassava products and many indigenous products. We have the LN mesenteroides, and then for bakery products like sourdough products and crackers, we have LB San Francisco, LB fermentum and so on. So, let us go into the second type of alcoholic fermentation, which is the alcoholic fermentation. This is a biological process where sugars are converted into ethanol and carbon dioxide by yeast, mainly Saccharomyces cerevisiae. This process is essential in making alcoholic beverages. We will be discussing about these a little bit more detail in the next lecture. We have similarly acetic acid fermentation. This is a process where ethanol is converted into acetic acid by Acetobacter bacteria in the presence of oxygen, essential for vinegar production. Then we have malolactic fermentation, which is a process where malic acid in wine is converted into lactic acid and carbon dioxide by lactic acid bacteria, primarily Enococcus oni. This fermentation typically occurs after the primary alcoholic fermentation. Then you have the butyric acid fermentation, which is an anaerobic process where bacteria, mainly Clostridium, convert carbohydrates into butyric acid, carbon dioxide and hydrogen in low oxygen environments like animal intestines and certain food. Human health importance is there of this butyric acid fermentation process. Butyric acid is vital for gut health, providing energy for colonocytes, supporting gut barrier function and preventing inflammatory bowel disease. And it also enhances flavor and nutrition in foods like cheese and fermented dairy products, and improves the nutrient bioavailability by breaking down complex carbohydrates and aiding in the absorption. It provides anti-inflammatory effect and reduce colon cancer risk, emphasizing the importance of dietary fibers for gut health. So, apart from these, we have mixed fermentations, which involves the simultaneous or sequential fermentation by different microorganisms such as yeast and bacteria. This method is widely used in producing various fermented foods and beverages. So, these mixed fermentation enhances flavor complexity. For example, in winemaking, the combination of wild yeasts and lactic acid bacteria can create unique flavor profiles that are more nuanced than those produced by single strain fermentation strategies. As this is mixed, this process promotes microbial diversity, leading to unique characteristics in fermented products. For instance, traditional sourdough bread often uses a mix of wild yeast and lactic acid bacteria, resulting in distinct taste and texture. Mixed fermentations improve the stability of the fermentation process. In beer production, the use of both yeasts and bacteria can help inhibit spoilage organisms, enhancing the safety and shelf life of the beer. And mixed fermentations produce beneficial compounds such as probiotics. For example, in yogurt production, the combination of lactobacillus and streptococcus species contributes to the health benefits associated with consuming yogurt. Now, let us look into some of the traditional fermented food around the world. We know the human civilization has a long history of fermentation, over 6,000 to 10,000 BCE old. Now, let us look what are the various kind of traditional fermented food available around the world. So, here you can see a kimchi, which is basically a Korean kind of a pickle. So, it has common ingredients which include napa cabbage, radishes, garlic, ginger and chili pepper, often combined with fish sauce or fermented seafood. This is made through a fermentation process where vegetables are salted and mixed with spices, then stored in jars to ferment at cool temperatures. And then there are regional variations within Korea and have unique recipes varying in spice levels and additional ingredients reflecting local taste and available produce, the raw materials basically. So, it has a lot of health benefits, provides probiotic effects, improve gut health, potential anti-carcinogenic properties. Then one of the popular fermented food is the soy sauce and miso in Japan, and this is produced by fermenting soybeans and wheat with specific molds and bacteria, followed by pressing and aging. Modern methods may use chemical hydrolysis for faster production. And miso is produced by fermenting soybeans with salt and the mold Aspergillus oryzae, varying in fermentation time and ingredients for different flavors and types. Both soy sauce and miso are essential flavoring agents in Japanese cooking. So, it impacts the Japanese cooking to a large extent. It enhances umami and contribute to dishes like soups, marinades and dressings. So, in Indonesia, there is a popular fermented food called tempe. You can see this being offered in a plate. It is made by fermenting boiled dehulled soybeans with Rhizophus oligoporus for 35 to 37 hours at room temperature, creating a soft chewy cake with a distinctive flavor. It has high protein, contains beneficial lactic acid bacteria and has reduced anti-nutritional factors, improving its digestibility and nutritional value compared to raw soybeans. So, it is a traditional Indonesian food, is a staple protein source deeply rooted in local diets and cultural heritage of Indonesia. In European countries also many interesting fermented food are made. For example, sauerkraut in Germany is made by shredding cabbage, fermenting it with 2 to 3% salt and utilizing natural microbial processes with leuconostoc and lactobacillus species, creating a tangy preserved product. And this is originated in Germany, and it has been valued for centuries for its preservation qualities and nutritional benefits, especially during winter months when fresh vegetables are not available. And it offers health benefits including improved gastrointestinal health, rich in vitamins and probiotics, although its benefits may not solely depend on live microbes. Then we have seeds which with many regional variations, not only in Europe but also other parts of the globe. So, you have numerous varieties like soft, semi-soft, hard and then blue seeds, each with unique flavors and texture. So, here you can see a feta seeds in this picture. So, this is white cheese made from sheep's milk or from a mixture of sheep and goat's milk. So, cheese production starts with selecting high quality milk, often pasteurized. Though raw milk is used for traditional varieties, starter culture and rennet form curds which are cut, cooked and drained off whey. The curds are then salted for flavor and preservation before aging, where the cheese flavor, texture and aroma develop. Microorganisms are crucial for seas characteristics. Penicillium roqueforti is the key for blue seases, while lactobacillus species contribute to acidity and preservation. So, there are many other regional fermented food. For example, here you can see salami, which is cured sauces consisting of fermented and air dried meat, and then we have here these fermented meat products typically include either pork or beef, or it can be any other meat source. Salts, spices and starter cultures often involving lactic acid bacteria for fermentation. Common methods include curing, smoking and drying, which do not only preserve the meat, but also enhance flavor and texture. Different cultures have unique fermented meat products, such as the salami in Italy, chorizo in Spain and bresaola in Switzerland, reflecting local taste and traditions. These fermented meats can offer benefits like improved digestion, enhanced flavor, although moderation is advised due to potential health risks associated with high salt and the fat content. So, like fermented fish, we have the fermented fish products, especially for traditional sauces like garum. So, here we can see the fermented fish product from Manipur. Fermentation is driven by microorganisms or enzymes involving acidification, protein gelation and lipid degradation, often taking several months. Fermented fish is significant in cultures across Southeast and Northern Europe, with each region offering distinct products and methods based on the local traditions and available fish species. Fermentation enhances the nutritional profile of fish, improves digestibility and produces antimicrobial substances that extend shelf life and reduce contamination risk. So, we have here the grass carp, the process for fermented jhayu product here shown. It is a traditional fermented fish product in China. So, here this is a grass carpet fillet which is cut, then these are finally cut into cubes, and then these are mixed with rice powder, salt, hot pepper, ginger and water, and then allowed for anaerobic fermentation by filling up with nitrogen and thereby making the oxygen unavailable, and the final product we get the fermented jhayu. In India also there are a lot of fermented food. We are unable to mention each and everything here, but this book Ethnic Fermented Foods and Beverages of India, Science, History and Culture 2020, which is edited book by Professor Tamang, is actually having so many different kinds of fermented food documented from India. Just mentioning here few, for example, bamboo shoot, right, which is actually when grown into adult is a very woody plant, but the soft shoot is actually fermented, and a product named Corisa, and it has many various different names, predominantly used by communities from northeastern states of India, as well as also some of the South Asian nations in the world. So, these are directly can be used as chutney, or it can become a ingredient for mashed vegetable or onion mustards, and you have these as a flavor enhancer in meat, fish, curries and pickles. And then there are various microbes involved, lactic acid bacteria, then we have Listeria. It produces bacteriocins inhibiting Listeria, monocytogenes, Staphylococcus aureus. Then another is the fermented oil seed, basically mustard seeds. So, that production is called as the karoli, served with rice meals or fermented rice, which again is a fermented fermentation of the leftover rice at night, and it is a overnight fermentation. It is a very complex microbial product. So, microbes involved are still to be characterized, and it is produced by various ethnic groups in Assam and adjoining areas. Let us now have a small discussion on the scientific principles of fermentation. Many of these are already mentioned to you in some way or the other. We will discuss about the microorganisms involved in the fermentation, the biochemical reactions that happens, and the environmental conditions that are required for fermentation. So, one of the important microorganism that is involved in fermentation is the bacteria. For example, lactic acid bacteria which is common in fermented foods, and it includes various genera like Lactobacillus, Lactococcus and Streptococcus, which help in the fermentation process by converting sugars into lactic acid, enhancing flavor and preservation. Bacillus species is another bacteria. This is found in alkaline fermented foods, especially in Asia and Africa. Species like Bacillus subtilis contribute to fermentation of legumes and produce L-polyglutamic acid, affecting its texture. So, here you can see here this Bacillus subtilis forming a visible structure with white dendrites in the nutrient gel dish within 2 days of inoculation here. Okay. And then here on the top you can see the Lactobacillus, which is a gram positive aerobic bacillus and used as a probiotic. Then we have the yeasts, for example, Saccharomyces cerevisiae, which is very common. Then we have Candida and Brettanomyces, which are prevalent in alcoholic beverages, bread fermentation, etc. And they play a role in alcohol fermentation, particularly Saccharomyces cerevisiae converts sugars into alcohol and carbon dioxide, essential for producing beer, wine and spirits. And of course, a solid food fermentation like bread is also a product of these yeasts. So, it ferments the dough, produce gases that causes the dough to rise, contributing to the texture and flavor of sourdough and other breads. So, here we have the Candida albicans fungi observed with an optic microscope, and here we have the Saccharomyces cerevisiae observed under a scanning electron microscope. Then comes the molds, and common genera includes Aspergillus, Mucor and Rhizopus, which are significant in the fermentation of various foods including cheese and soy sauce. Molds contribute to the development of flavor and texture in cheeses, aiding in the breakdown of fats and proteins, which enhances the overall quality of the cheese. Molds like Aspergillus are crucial in the initial fermentation stage of soy sauce, where they help convert soybeans and wheat into sugars, which are then fermented by yeast and bacteria to produce the final product. So, here we can see an image of a Aspergillus species, and this is Mucor, a genus of about 40 mold species commonly found on cheese like Tomedi Savoy. Then we have certain biochemical reactions happening when the food is getting fermented. For example, there is the conversion of the carbohydrate. So, glycolysis is the initial pathway in carbohydrate fermentation, where glucose is broken down into 2 molecules of pyruvate. This process occurs in the cytoplasm and generates a net gain of 2 ATP and 2 NADH molecules, which are essential for energy production. In the absence of oxygen, pyruvate undergoes fermentation to produce ethanol and carbon dioxide. This process involves 2 key steps. One is the decarboxylation. Pyruvate is converted into acetaldehyde by releasing carbon dioxide, and then the reduction of acetaldehyde to ethanol using NADH, which generates NAD plus needed for glycolysis to continue. Then we have the lactic acid fermentation of carbohydrate conversion, where pyruvate is directly converted into lactic acid. This process is common in muscle cells and certain bacteria, allowing for energy production without oxygen. So, here we can see sugar metabolism by Lactobacillus and Saccharomyces as representatives of lab and yeasts. So, you can see in figure A the fermentation carried out homofermentative pathway and the heterofermentative pathway, where in the first glucose is converted to 2 lactic acid, and then in the heterofermentative pathway glucose is converted into ethanol. And in the case of Saccharomyces also, you can see how glucose gets converted into 2 molecules of alcohol. Let us have a small discussion on the production of byproducts like the production of acids. Fermentation produces various acids like lactic and acetic acid, which contributes to the sour flavor of fermented foods and aid in preservation by lowering pH, thereby inhibiting spoilage organisms and pathogens. And it also produces gas like carbon dioxide, which is a key product and plays a crucial role in bread making by causing dough to rise and adding effervescence to beverages, enhancing texture and the mouthfeel. Then we have the alcohol production, Ethanol is primarily produced during the fermentation of beer and wine. It adds flavor and acts as a preservative due to its antimicrobial properties, adding the shelf life of fermented products where it is produced alongside. Then we have the acids, gases and alcohols generated during fermentation, which enhances the flavor profile of foods and beverages, creating a range of flavors from fruity and floral to sour and tangy. Now, let us have a small discussion on the impact of environmental conditions on fermentation and product quality. Mostly temperature, pH, salt, oxygen plays a very crucial role. For example, most microorganisms thrive at 20 to 35 degrees centigrade, but higher temperature may cause off flavors or harm them. And again, lower temperatures may actually slow the fermentation process. Then we have under pH variations, something like say a slight acidic pH will support fermentative bacteria, and it will also prevent spoilage. So, because it will not allow those spoilage microorganisms to grow at a slightly acidic pH. Salt is very crucial. It enhances flavor and inhibits harmful bacteria, but excessive salt can slow down fermentation. And the presence and absence of oxygen is also very important environmental condition. Anaerobic conditions are essential for alcohol fermentation. Some lactic acid bacteria can ferment with or without oxygen. So, with this we come to end of this lecture. Thank you for your kind attention.